Hello friends and dear students, welcome to my science, biology and biotechnology classes. Today in continuation with biomolecules, I am going to discuss about protein and basic unit of proteins, that is amino acids. So all of you know what are the total different type of amino acid present in your body, that is 20. So what you say a standard amino acid, what do you mean by a standard amino acid? A amino acid which is coded by codon. You know codon is triplet, commaless, universal, unpunctuated. So that is one codon is responsible for one amino acid. If you go through the biochemistry, then AUG is initiation codon that will code methionine. Okay. In this way, there are 61 codon that will code different 20 standard amino acids. There is some amino acid that is coded by more than one codon. So that is degeneracy of the codon. So let us try to know what do you mean by a standard amino acid. Means amino acid coded by particular codon. Then we can say it is a standard amino acid. Next, in category of standard amino acids, there are two types. One is essential amino acids and other is non-essential amino acid. What do you mean by essential amino acid? An amino acid which is going to synthesize in the food we cannot synthesize in our body and we are taking through our diets. Then we can say it is essential amino acid. How can you learn whether this is essential amino acid or non-essential amino acids? So there is one trick like treat very ill MP at hospital. Just for learning, I'm just telling you, just try to remember, treat very ill MP at hospital. Means T, that is T denotes for tryptophan. This is tryptophan. And then that T, that is threonine. Okay. And that V, that is alanine. And then I denotes for isoleucine. And then this, that is leucine. And again that one, that is lysine. Okay. Then MP, M means methionine, and then FP means phenylalanine, and then A means arginine, and then H means histidine. So this type of question frequently asked in so many competitive exam or so many CBSE based exam. List out 10 essential amino acid. Rest all other are non-essential, means that can synthesize by our body. That is non-essential amino acid. No, that is synthesized by our body. Then there is no need to take from outsource, means through food. So treat very ill MP at hospital. T4 tryptophan, T4 threonine, G4 valine, I4 isoleucine, L4 lysine, L4 lysine, M4 methionine, P4 phenylalanine, A4 arginine and H4 histidine. Among these 10, these two are semi-essential. Arginine and histidine are semi-essential amino acids. Okay. So try to know what is protein. Protein is macromolecular. That is a smaller size. Linear polymer. It can be linear polymer. It can be branch by joining other bonds. High molecular, organic in nature, organic weight compound joined together by peptide bond. Peptide bond. Okay. So we can say it is a protein. Okay. So all of you try to know there is a protein, there is a polypeptide. So we cannot say all the polypeptide it is a protein. It should be minimum 100 or 1000 amino acid joined together. Okay. Now try to know among these 20 amino acid, just I am going to discuss about a structure, chemical structure of amino acid. The first amino acid is glycine. So glycine is the simplest amino acids. Why it is simplest? Because there is less number of carbons. You can observe here. Here, how can you recognize whether it is the carbohydrate or it is the protein amino acid? Just to present the chemical bonds or chemical groups. Okay. So mainly on the right side, there must be acid group on the first carbons, and then on the left side you will find amino groups. Amino groups. Okay. And above you can have to write hydrogen. So these three groups must be present in any standard amino acids. Means acid group, COOH, then hydrogen, then amino groups. And you will find there is R. R means alkyl. Hydrocarbon chain you can observe. But although this glycine is simplest amino acid, so there is no any carbon along with hydrogen. Only hydrogen is present. So that's why we can say it is simplest amino acid. So how can you learn to draw the structure of amino acid? Simple. You can observe, if you remove this hydrogen from the glycine, then if you add one methyl group, you know methyl and methylene, this one is methylene groups and this is methyl groups. This is methylene groups and this is methyl group. So methyl group must present on the terminal end and methylene group present between the chain. So if it is added by methyl groups by replacing the hydrogen, the same structure you can observe, nothing change. up to that point. You can say it is the head of the amino acids. It will present in all the structure where it will change in R group, that is alkyl group. In case of biochemistry or chemistry, if we are going to remove hydrogen and add methyl group, then we can say it is alanine. Simple. You remove that and you add. So this is alanine. Now come to next amino acid. What you have to do? Same structure, it will remain there. Just see up to that point, it is same structure. What you have to add? Simple 2 carbon along with hydrogen. Means here you can change by putting 2 methyl groups. Then I am going to convert this alanine into that valine. Just try to compare. So initially it was alanine, but you know there is CH3 and there is 3 bonds. So you have to remove the what? You have to remove hydrogen. 1, 2, 3 and 1 hydrogen. Okay? Just to fulfill the valency of carbon. Okay? So this total structure, that is a valine. You can find a branch in alkyl groups in case of valine. Okay? Next, you can also observe that if you derive or lysine, if you derive or lysine, then you have to do slight change. What change? You can compare. Just see this structure, it is common. It is common in all amino acid structure. That is the head of the amino acid chemical structure. Now, this is also present in valine. What extra carbon and 2 hydrogen. So that group means 1 methylene group. Simply if you add above the branch, then that will be the lysine. So that structure will be the lysine structures. If you observe in your protein or structure of that different amino acids. Now, if you see, try to compare and then you can learn. Means, in this case, either you count the number of carbons or you count the branch chain R groups. On that basis, slowly and slowly, you can derive the different amino acid chemical structures. So these all are alkyl group containing amino acids, and you know other amino acid, that is isoleucine. Okay, so you just try to see what type of shape it is. That is T shapes. In case of valine, you will find this shape, inverted Y shape, and again in this case lysine, you will find again that shape on, means that carbon, 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 and then this is also carbon. In this case there is more carbon, in this case there is less carbon, but here you can observe there is a T shape, means a branch in alkyl groups is there. You know this is the head. This is the head of isoleucine. In every amino acid you will find this head, that is 1 acid group, 1 hydrogen, 1 amino groups, like that. But you simply draw a T-shaped structure like that. Suppose this is T, and you indicate this is the carbon point. Okay, this is the carbon point. So one carbon, one carbon, one carbon, and one carbon, and one carbon. So simply add that in the alpha carbons. Why it is alpha? You know, in nomenclature, in chemistry or biochemistry, this is the top priority groups in chemistry, organic, especially in organic chemistry. After this carbon, whatever the carbon, that is known as alpha carbon. So you have to add a T-shape, a structure of carbon chain here. So that you have to add again CH3 and again CH3 and then CH2 like that, and here also you can add CH3. So either we are going to add methyl group or methane group in case of bulky R groups, in case of just to derive different type of amino acid structures. Okay. Now come to different other amino acids. Like if you see aromatic amino acid, if you see aromatic or hydroxy group containing amino acid, try to hydroxy group containing amino acids. Yes. So what you have to derive in case of hydroxy group containing amino acid, that is serine and threonine. Serine, you know serine. So you first of all try to draw the first head. That is, this is acid group. Always you have to show acid group on the right side, and then hydrogen group, okay, and then amino group, and then 1, 2, 3, and 4. Okay. So just count the number of carbons, and you have to remember that. Then you have to add here methylene group, and then you can add this hydrogen. Hydrogens. So try to understand what is the total number of carbons in this case. That is 4. 1, 2, 3, 4. Okay. And you can add one hydroxy group. So that amino acid is known as serine. Okay. Serine, this is the particular amino acid, that is one hydroxy group. Okay. Next amino acid, that is threonine. Threonine. You know, you have to add simple acid group and then one hydrogen and one amino groups, and again you can draw the similar structure, that is 1, 2, 3, 4. 1, 2, 3 and 4. Okay. So what is the difference between serine and threonine? Simply you can add a hydroxy group. Where hydroxy group you have to add? Where the hydroxy group you have to add? In this carbon. Okay. And then simply you put hydrogen and then hydrogen. So this is the structure of threonine. This is the structure of threonine. That is the hydroxy group containing amino acid. Next, that is sulfur containing amino acid. Sulfur containing amino acids. Okay. Sulfur. You know, sulfur is very, very important. Sulfur containing amino acids. Same things. You have to draw like hydroxy group containing amino acid. This is H and this is H and this is NH2 and there is CH2 and then you have again CH2 and then SH. Okay, so you know why I am removing that two hydrogen, because you know if you add this, you have to add, okay, this hydrogen, okay, and then you can draw like this one or this one, okay. So that is SH group, thiol. You can say it is thiol group. So the particular amino acid, that is cysteine, is sulfur containing amino acid. How many amino acid carbon is there? 1, 2, 3 and 4. Then other amino acid that contains sulfur, that is methionine. Methionine. Okay. So what you have to do? Simply you have to add one CH3 group here and one carbon here. One carbon here instead of hydrogen and one carbon here. And so you have to add here methylene group and here methyl group. So draw like that. COOH, then hydrogen, then NH2, and then you can draw like similar structure. CH2, okay, and then CH2, okay, and then what you have to add? One more CH2 and then S and then CH3. So how many carbons is there? 1, 2, 3, 4. In this case they have 4 carbons. But if you draw the structure of methionine, what? Cysteine and methionine, both are thiol group, means sulfur containing amino acid. So just go through this chemical structures, that is, you simply add one CH2 here and you have to replace this one and then you have to add CH3. Okay, so like that. Okay, just to convert this cysteine into methionine. Okay, so just see here, I have added this one group, that is methylene groups. This is methylene groups, and at the end I have added methyl groups. Methyl group. So that is thiol group, that is sulfur containing amino acids. Next amino acid. Which amino acid is acidic in nature? Which amino acid is acidic in nature? So try to draw acid group containing amino acid. Acid group containing amino acids. So let's start. Simply you have to draw a same repeated structure, that is COOH and then hydrogen and then amino group and then CH2, then more CH2, okay, 1, 2, 3, and you know this is acid group, so that is OH, that is acid group containing amino acids. So that is aspartic acid, that is known as aspartic acids. This is aspartic acid. There are two amino acid which contain acid group, that is aspartic acid, and other is glutamic acid. So what is the difference between aspartic acid and glutamic acid? Only one methylene group if you add. In this case, then it converted into glutamic acid. So why I am telling this is aspartic acid? Why not aspartame? Acid group is there. You know, this is acid group. You will learn in this organic chemistry. or biochemistry, this is acid groups. There is one more acid group. So that's why it is acidic amino acid. Okay. Now, if you want to draw the chemical structure of glutamic acid, then simple you have to repeat the same thing in more times. Okay. That is acid group, and then CH2, and then one more CH2, and then C double bond O, and then O and H. Okay. So that whole structure we can say it is acid. Okay. So try to differentiate what is the difference between aspartic acid and glutamic acid. That is acidic amino acid. So question asks, which of the following are the acidic amino acids? You will get a choice: aspartic acid, then leucine, then valines, and then isoleucines. So where there is acid word at, you can also say it is aspartate, and then the same meaning is there. Means aspartic acid also known as aspartate, and glutamic acid glutamic. Okay. So if the acid group is there, then the name automatically come in at the end suffix, that is acid. So these are the aspartic acid and glutamic acid chemical structure. Next, amide group, CO and H2 group. CO and H2 group containing amino acid, that is amide group containing amino acid. The question will ask like that. Okay. So the amide group containing amino acid, you can say the amide group containing amino acid. Which are the following are amide group containing amino acid? Then you have to write the name like that: asparagine, and other is glutamine. So such type of question may ask, which of the following are amide group containing amino acid? Then you have to write, observe the choice: asparagine and glutamine. If you have learned the aspartic acid structure and glutamic acid structure, the same structure is there. There is only slight difference. What is the difference? Instead of hydroxy, there will be NH2 and H2 group, CO and H2. Here you have also removed the hydroxy group, and then you have to add the NH2. So this is the total structures are similar like aspartic acid, glutamic acid, asparagine, glutamines. Asparagine contain amide group containing amino acids. So you have to learn this type of chemical structure, and you can draw easily if you draw once and you practice that. That is the important, because such type of question frequently asked in any competitive exam, either medical entrance test or engineering entrance test, like anywhere, from the chapter biomolecules. Okay. So this is amide group containing amino acid, that is asparagine and glutamines. Now I am going to draw the chemical structure of what? That is aromatic amino acid. You know, aromatic amino acid, very important. You know, that is aromatic amino acid. So what is aromatic amino acid? That is, you will find, you know, this structure, that is phenyl groups. Okay. If you observe, this is linear structure. So why it is ring? Because there is a delocalization of electron. You will find alternate double bonds. Alternate double bond. So it will shift and show resonance structure. It will shift and it will show a resonance structure. What is resonance? Delocalization of electrons. Delocalization means it will, you cannot say that is the fixed position of electron. In one time you can observe this type of bonding, in other time you can observe this type of bonding, means alternate double bond. You can observe in that ring, that is benzene. Okay, so that is phenyl. Okay, so here there will be carbon, 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 carbon like that. Okay, and then hydrogen you can add, hydrogen, hydrogen like that. Okay, so I am just trying to draw the aromatic amino acid. So that aromatic amino acid, simply you have to learn a head of the amino acid. What is head? You know, simply that is the head of amino acid. Okay, and now you have to add one CH2, and then you have to draw like that. Okay, so this is phenyl groups. This is of phenyl groups. Now you can draw a alternate double bond. What is the alternate double bond? One carbon, one carbon, one carbon, one carbon, one carbon, one carbon. So if you are drawing the any chemical structure, you must see whether the carbon valence is fulfilling or not. In this case, you will find 1, 2, 3, 4. So it is correct. 1, 2, 3. So you have to fill with hydrogen. Okay. Then it contains 4 valence. 1, 2, 3. Then you have to fill hydrogen. 1, 2, 3. Again the 3 valences. So you have to fill by hydrogen. Again here 1, 2, 3. So you have to fill hydrogen. And then 1, 2, 3. So you have to fill with hydrogen. So that is phenylalanine. That is phenylalanine. Okay. This is phenylalanine. Just I am going to draw a chemical structure. Just alanine also contains that type of structure. So only instead of last carbon you will find a phenyl. This is phenyl group. This is phenyl group. Okay. Somewhere you will find like that. Okay. This is a ring structure, means delocalization of electron will be moving around like that. And next structure, that is tyrosine. Tyrosine. Okay. So tyrosine, what is the difference between phenylalanine and tyrosine? Simply you keep in mind, you have to repeat that structure. Okay. That is nitrogen and then hydrogen, hydrogen and hydrogen, and you will find CH2. So what you have to do? Simply you have to add one more hydroxy group here in this points. Okay. So just try to draw this phenyl groups. Okay. So just give alternate double bond and then carbon. Yes, this is also carbon, this is carbon, this is carbon, and this is carbon. Okay, and this is also carbon. Okay, so you have to fulfill the valency with hydrogen, hydrogen, hydrogen. Okay, hydrogen and hydrogen. So you have drawn the same structure like phenyl, but you have to replace this hydrogen with hydroxy group. Simple. If you draw a phenylalanine, then you have to remove the this is the ortho and this is meta and this is para position. So you have to remove the hydrogen from the para position of the phenyl groups. That's why it is known as tyrosines. So this is chemical structure of tyrosines. Okay. Next you have to draw tryptophans. You have to draw the tryptophan. I have drawn the phenylalanine, that is aromatic amino acid, and then tyrosine, that is other aromatic amino acid. And now I am going to draw the structure of phenylalanine. Okay, so just simple, it is also simpler. In this structure you can draw like that, means turn like 90 degree. Okay, then you will find the structure of tryptophan. The tryptophan contains maximum carbons. You will find, in case of tryptophan, you will find the maximum carbons. Okay, so now in this time I am going to tilt this structure like that. Okay, now if you tilt that one like left anti-clockwise direction, this portion, I am just tilting this in anti-clockwise direction like that one. So that will come in upper position like COOH. Yeah, you can show like that, COOH. Okay, and then this will come at the bottoms like NH2, and then this hydrogen will be there in left side. Okay, and then you have to draw, you have to add one methylene groups. So this is methylene groups. No, you have to draw one bond also, otherwise it is incorrect structure. So try to understand, this is methylene group and this is methyl group. You cannot show any bond here. So this is methylene groups, and next you have to draw pentagons. You know pentagons. This is a pentagons. Okay, and then hexagon. You have to show one hexagon. Okay. So this is chemical structure of tryptophan. Okay. So in case of tryptophan, you will find there is a carbon, and there is a carbon, and there is a carbon, and there is a nitrogen. So in this tryptophan ring, that ring is known as tryptophan ring. Okay. So in this case, if you observe, there is a bonding. Double bond. Okay. Now you have to show a carbon. It is already fulfilled, and then you have to draw alternate double bond here and then here. Okay. So this is tryptophan ring. This whole structure is known as tryptophan ring. Now check whether carbon valence is fulfilled. 1, 2, 3, 4, it is okay. 1, 2, 3, 4, it is okay. And then you will draw 1, 2, 3. Only 3 valence is there. So you have to draw one more hydrogen. Okay. Now this is also carbon. So you have to draw 1, 2, 3. The 3 valency of carbon. So you have to fulfill the 4 valency with hydrogen. Okay. And then again 1, 2, 3. So you have to draw one more hydrogen, and 1, 2, 3. So there is also carbon. So you have to draw one hydrogen. In this case also 1, 2, 3, 4 already valence is fulfilled. Now nitrogen has 3 valency. So you have to draw one more hydrogen. Now you can see 1, 2, 3. So you have to draw one more hydrogen. So it is complete now. So this is the structure of tryptophan. That tryptophan amino acid has contained maximum number of carbons. Count it. 1, 2, 3, 4, 5, 6, 7, 8, 7, not 7, 7, 8, 9, 10, 11. Okay. So you have to count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So maximum number of carbon is present in this tryptophan, and that contains tryptophan ring. So you must try to recall it. What are the aromatic amino acids? That is phenylalanine, tyrosine, and tryptophan. I will discuss the basic amino acid in next day. Before that, I want to discuss a bond, chemical bond. That is very important between two amino acids. So what type of bond is there? You know, you will find, suppose I am going to draw a chemical structure of two basic amino acids, that is simple amino acid. Simple amino acid, that is, suppose I am going to draw one simple amino acid. Just this is acid group and this is hydrogen, this is hydrogen and this is NH2 group. Okay, so this is glycine. Okay, and next I want to add this other amino acid like COOH alanines. So you will add here CH3 group and then hydrogen and then nitrogen amino groups. Okay, so if there will be reaction occur, then this will combine. Okay, so this will combine and release water molecules. What it will release? Water molecules. Okay, so whatever the chemical bond form, that is peptide bond. That is peptide bond. Okay, so I am adding two amino acids, so it is known as dipeptide. On the basis of number of amino acid, we can say it is a dipeptide or tripeptide. If the 3 amino acid will join, if the 4 amino acid will join, then tetrapeptide. But number of peptide one will be less. Okay, if you count it, in case of dipeptide only one peptide bond is, in case of tripeptide 2 peptide bond present. So this is chemical structure, try to understand. Okay, so this is, you know, just try to know, that is N and H. Okay, and then this C and then hydrogen and then CH3 and then COOH. Okay, and now try to learn. This is carbon, that carbon, and you will find this is hydrogen and this is hydrogen and this is NH2. In case of any polypeptide chain, general convention is that in left side you will find amino group, on the right side you will find the acid group. You will find the acid group, and this is the peptide bonds. This is the peptide bond. Okay, so this carbon is C alpha. This is C alpha, C N and C alpha. Okay, so why it is C alpha carbon? Because just after acid group that carbon will present, that is known as the alpha carbon. There is C alpha, then C, and that is where it is, and where that is N, not H. It is N. Okay, and then C alpha. So you will find the rotation of only this bond. There is rotation of only this bond and this bond occur. Means this bond can rotate and this bond can rotate. Rest other, it will remain in the one plane. It will not rotate. That bond will not rotate. So this will remain in whole plane. That is in whole plane. So it is remain in one plane. So you will find this is CO and this is N, H, CO and H. So that whole structure is known as peptide bond. Okay. So you try to know what is peptide bond. A peptide bond that is formed between acid group of the first amino acid 1, and this is amino acid 2. Okay, and the amino group of the second amino acid. Generally acid group present on the right side and amino group present on the left side. So acid group of the first amino acid react with amino group of the second, and then reaction will occur after release of water. 1 molecule of water, you will find this chemical structure, that is peptide bond, and all will remain in one plane. I am talking one plane means all the atom will stick in one plane. If it will rotate like that in one plane. But that bond can rotate. Okay, that bond can rotate. Means C and C bond will rotate, N and C bond between N and C will rotate, but this will not rotate. It will rotate along with the plane. Okay, so that will show the resonance structure also. That will show the resonance structure also. So that's why it is delocalized. Okay, so that type of bond between two amino acids, that is known as peptide bond. Okay, so try to understand what is the simplest amino acid, that is glycine. What is the branching containing R group containing amino acid, then it is known as valine, then leucine, then isoleucine. This is valines, then leucines, then isoleucines, and then sulfur containing amino acid, that is cysteine and methionine. Hydroxy group containing amino acid, that is serine and threonine, and acid group containing amino acid, that is aspartic acid, glutamic acid, and the amide group containing amino acid, that is asparagine and glutamine. Okay, and aromatic amino acid, that is phenylalanine, tryptophan and tyrosine. And what is the peptide bond? Bond between two amino acids after release with water. So that is the simple structure of the different amino acid, and this is the head group. You have to remember always, it will must present on the head region, that is alpha carbons, must surrounded by different groups, that is asymmetrical. Only one amino acid that do not contain chiral carbon, that is symmetrical carbon. Why? Because both the group are similar. So this is the exception. Rest other amino acid contain chiral carbon, that is asymmetrical carbon. Such type of question may ask in your competitive exams and in your coming CBSE exams. So try to remember what are the different group present in the different amino acid. Rest basic amino acid I will discuss in next class, and some structure like primary structure, secondary structure, tertiary structure I will discuss in next class. So thank you and have a nice day.